Hey guys, it's Miss Pham. These notes are for DNA. We're going to go on page 41 and 40. On 41, we're going to talk about the function of DNA, what does it do, what is it made up of, and then we're going to talk structurally about DNA on page 40. Okay, so getting started, again, we're talking about DNA on page 41. DNA stands for deoxyribonucleic acid. That's where it gets its name from, the D and A. I have already underlined it in your notes for emphasis, D and A. DNA is present in the nucleus of eukaryotes. Now, eukaryotes are animal cells, plant cells, fungi cells. They all have nucleuses. They have other organelles as well. Now, but a prokaryote is like a bacteria. The bacteria don't have nuclei. So their DNA is in their cytoplasm. It's kind of free-floating around. Alright, so its function, very big here. Oops, sorry. Its function is to control or to hold on to genetic information and control what happens in the cell by making proteins. Again, so your keywords are hold genetic information, control, and make proteins. Big key here. That's the whole point of DNA, is that it codes for proteins. It gives the instructions on how to make proteins. Proteins carry out all living functions in your body. There are structural proteins that help change the color of your eyes or set the skin color on your body, and there are proteins that help you digest foods, like amylase, for example, is a enzyme, which is a type of protein in your saliva that helps break down sugars as you chew and swallow. Shape of DNA is considered a double helix. Double helix is a fancy word for a twisted ladder, so I kind of drew one right here. It's two squiggly lines kind of wrapping around each other. And between them, we say twisted ladder because there are, it looks like steps inside. Now, that is the general shape of DNA. Then what it can do is it can be wound up real tight into chromosomes. You'll hear this word a lot as we go forward and we talk about cell division and cell growth. Okay. This double helix shape was founded by a pair called Watson and Crick. There are many other people who have also contributed to the shape and the understanding to DNA, but those two are the ones that we focus on. Alright, DNA is a type of nucleic acid, just like RNA and ATP. You can go back into your notes if you're really unsure about nucleic acids. I think on page 7 should be a chart of what nucleic acids are made up of, what are their functions, all that good stuff. What you need to know is that because it's a nucleic acid, it's made up of nucleotides. That's the building block. A nucleotide consists of a phosphate, a sugar, and a nitrogen base. There's your phosphate, the circle, sugar, your pentagon, and your nitrogen base is the rectangle. Type of sugar it uses is deoxyribose. Surprise, surprise. That helps it name the molecule, deoxyribonucleic acid. Types of nitrogen bases make two broad categories, purines and pyrimidines. Purines are your A and your G. Pyrimidines are thymine, cytosine, T and C. Now, they're categorized based on their chemical structure, which we won't get into, but just understand that there are two categories, purines versus pyrimidines. Alright, so this is page 41. We've talked about what it stands for, where can you find it, what is its function, its shape, what is it made up of. Now we're going to go back to page 40. and you're going to talk about its structure. Alright, I'm not going to go through how to draw the shape in the center right here. If you have any questions about that, come see me before or after school. It was a pretty long process for us to get that down, and I'm just going to go over some of the things we wrote on the side and explain the parts of the DNA structure. So let me erase this centerpiece. I'll erase everything actually. Let's give us a fresh start. Alright, so first things first, your DNA chains, they're two linked together. That's what makes the twisted ladder. One runs, let's say, north, and the other one runs south. All you need to understand is that they run opposite each other. The opposites allow for the nitrogen bases to pair up and attach. Notice I actually wrote the letter upside down so that you could really understand that it is a different direction. Okay, now let's talk about the twisted ladder shape. A twisted ladder starts off with two really strong sides. Those are your bases. So your base or your backbone is a repeating chain of phosphates and sugars. Phosphate, sugar. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. You know, repeating, so on and so forth. That makes the base of your ladder. Alright, between your long foundational backbones has to be your steps. You can't go up a ladder if there are no steps for you to stand on. So your steps are made up of your nitrogen bases. See how they run across. Nitrogen base, nitrogen base, nitrogen base, nitrogen base, nitrogen base. Okay? So we've already talked about sugars attaching to the phosphates in long chains. That the chains run in opposite directions. Nitrogen bases pair up to link the two chains together. Now, last thing you need to know, and as you go forward, you'll understand that certain nitrogen bases pair up with particular nitrogen bases. So a kind of generalization is purines always pair up with pyrimidines. So if you go back to page 41, or you just look at page 41, you can see that purines are A and G. So A and G are never going to bind, and C and T are never going to bind. It's going to be across the table. So A is going to pair with T in DNA, and G with C. Again, we'll go over more of what pairs with what, but I wanted you to understand its general function, its structure, and what it's made up of. So that's DNA. If you have any questions, go back, skip parts that you understood, and just focus on what you need to for the test. Alright, see you in class.